Okay, so it's a pleasure for me to introduce this week's seminar speaker, Nikita Finger. And Nikita did a BSc at the University of Central Florida, and then a BSc and an MSc at the University of Cape Town. She completed a PhD at the University of Cape Town under the supervision of David Jacobs and Marc Holderied from the University of Bristol. The PhD was submitted last year and she's due to graduate in July. Her thesis was about investigating how changes in the echolocation pulse parameters of horseshoe bats allowed them to occupy a wide varieties of biomes. And I'm really looking forward to hear the talk today. So let me just stop sharing for a second and I'll get Nikita to quickly say hello to you before I'm going to move to showing you the pre-recorded presentation.
Hi everyone, my name is Nikita and I hope you enjoy my talk today.
Thank you, Nikita. So I'm going to start sharing and starting the presentation.
Hello everyone. So today I'm going to take you on a journey of how habitat affects the acoustic properties of bat echolocation calls, and how this in turn shapes how these creatures detect prey in their different environments. Bats are excellent nocturnal hunters, and this is largely in part to the utilization of sound for orientation and foraging, a system known as echolocation. Bats echolocate by emitting a high-frequency sound, and based on the properties of the returning echo, the bat can tell the size, distance, and texture of objects in its surroundings. For the purpose of this presentation, I'm going to focus on echolocation calls that have a constant frequency modulated structure, also known as high-duty cycle echolocation. High-duty cycle bats separate their echolocation calls from their echo in frequency rather than time. The constant frequency component, along with Doppler shift compensation and acoustic lengths, is used to detect the fluttering of insect wings against background vegetation, making them particularly well suited to foraging in high cluttered environments. The frequency modulated components are thought to be important for ranging and potentially communication. I'm going to explore how habitat correlates with pulse parameters that determine detection distance. Detection distance is the distance at which a bat can detect its prey. This is in turn determined by the call's acoustic properties and the habitat in which the sound is made. The two acoustic properties that determine detection distance include call intensity and call frequency. Frequency is the number of wave cycles that occur in 1 second. So high frequency sounds have short wavelengths, while low frequency sounds have long wavelengths. As a result, high frequency sounds capture more detail, but they don't travel as far as low frequency sounds. Intensity also plays a key role in how far a sound travels. The intensity of a sound is the extent to which air particles are displaced. High intensity sounds have high amplitudes, which means they have higher changes in air pressure as the sound wave passes than low intensity sounds. The more intense the sound is, the farther that sound travels and the louder it appears. So for example, children whispering would be low intensity, while lions roar would be high intensity. So for this reason, animals that use high frequency sounds, which don't travel very far, such as the echolocation of a bat, tend to use high intensity sounds. It's important to note that intensity is energetically costly. As peak call intensity rises, so does metabolic rates in bats. Above 130 decibels, Atmospheric conditions such as temperature, relative humidity, wind, and barometric pressure result in the degradation of sound, while clutter contributes towards the absorption of sound. In cluttered environments, bats do not need to detect prey at as great of distances as in open environments. For my project, I investigated the foraging habitat hypothesis, and this proposes that habitat has a major effect on the echolocation parameters of bats. In particular, I looked at how habitat affects frequency and intensity, and therefore detection distances of bats in different biomes. Peak frequency is defined as the frequency at which a bat puts most of their call energy into. Intensity was measured 10 centimeters from the bat's mouth, also known as emitted source levels. I tested the foraging habitat hypothesis by looking at the echolocation parameters of one species in particular, Rhinolophus capensis. This species occurs in different biomes along the southern coast of South Africa. Gene flow has been established between these different populations, so we're controlling for phylogenetic history. In addition, both mitochondrial DNA and nuclear introns place the populations as belonging to the same species. But what makes this species ideal for testing the foraging habitat hypothesis is that it echolocates at different frequencies in these different biomes. So Odendahl et al. Investigated this phenomenon, and she found no correlation between atmospheric conditions and variation in frequency. But she did find a positive correlation between level of clutter, measured here as NDVI, and frequency, with lower frequencies occurring in lower degrees of clutter. So to explore whether these lower frequencies resulted in greater detection distances in these open biomes, we calculated the intensity and detection distances of two populations of Archipensis. Archipensis in the desert that echolocated 75 kilohertz, and Archipensis in the fynbos that echolocated 84 kilohertz. As an additional test of the foraging habitat hypothesis, we also explored the call intensities and detection distances of Rhinolophus damarensis. Now, Rhinolophus damarensis occurs in the same desert habitat as Archipensis, but it echolocates at a frequency that's equivalent to Archipensis in the fynbos. According to the foraging habitat hypothesis, differences between desert and fynbos bats are the result of ecological selection for increased detection distances in the desert in response to lower degrees of clutter and therefore increased volumes of space. So the following predictions can be made. Detection distances for bats inhabiting the desert should be similar to each other, but greater than those of bats inhabiting the fynbos. In the desert, R. damarensis should use higher intensity calls than Archipensis, essentially compensating for the use of higher frequency calls and achieving comparable detection distances. And lastly, if differences in frequency offer increased detection distances, as proposed by the foraging habitat hypothesis, then differences in detection distances between Archipensis in the desert and fynbos should largely be due in part to differences in frequency rather than differences in intensity. Methods were as follows. We recorded all species of bats in the field during emergence and at foraging sites. To calculate intensity, you have to know the position of the bat in relation to the microphone. To do this, we used microphone array analysis. Microphone array analysis works as follows. So in the diagram, you have one array with 4 microphones. A bat produces a call that radiates outwards and is recorded with a slight delay by each one of the microphones. The time of arrival differences of the call to each of the microphones is then calculated using cross-correlation analysis. The position of the bat in relation to the microphone is then triangulated and produces what is known as the bat's acoustic flight path. The environment was then mapped out using a laser distometer, creating a 3D scan of the environment. 3D scans were overlaid with the bat's acoustic flight path so we could visualize, quantify, and compare clutter in which the bats were flying at each recording site. So if we go back to our stipulations of the foraging habitat hypothesis, we determined whether or not prey detection distances for desert bats were similar to each other, but greater than feignbos bats. And if so, did Ardamarensis compensate for the use of higher frequency calls by using higher intensity calls? So as you will see, in the desert, Arcapensis not only used lower frequency calls, but they also used higher intensity calls, resulting in significantly higher prey detection distances for small, medium, and large prey. In addition, within the desert itself, Arcapensis emitted high intensity calls, greater detection distances when flying in a foraging area versus when emerging from a cave. However, the intensities and prey detection distances used by Ardamarensis in the desert tell a different story. Ardamarensis in the desert had lower average detection distances than Arcapensis in the desert. In fact, they were similar to those used by Arcapensis in the feignbos. And they didn't compensate for higher frequencies with higher intensities. However, this is not the end of their story. So if you go back and you look at the intensity ranges used by both Arcapensis and Ardamarensis, you'll note that Ardamarensis uses both much lower and higher maximum intensities than Arcapensis, and that by using higher call intensities, Ardamarensis was actually able to achieve detection distances greater than or equal to those used by Arcapensis in the desert, despite their use of higher frequency calls. So before we look at what these results could mean, let's look at our last prediction. So we predicted that if differences in frequency between feignbos and desert Arcapensis are indeed for increased detection distances, as proposed by the foraging habitat hypothesis, then differences in detection distance should largely be a result of differences in frequency rather than differences in intensity. To determine this, we did an analysis where we calculated detection distances in the desert when keeping one acoustic parameter constant, so either frequency or intensity, and using the averages of the second acoustic parameter from each of the biomes. And then we compared these differences. So when you look at just lower frequency calls, you do get a significant difference in detection range. However, this difference is almost doubled when you just use higher intensity calls. So therefore, intensity does play a greater role than frequency in achieving greater detection ranges in the desert. However, the greatest effect was seen when using both higher intensity and lower frequency calls. So in conclusion, Damarensis could compensate for their higher frequency echolocation calls by emitting high intensity calls, allowing them to achieve similar detection distances to Capensis in the desert. However, on average, they did not emit these high intensity calls, and this resulted in
Estoy en una reunión, no te oigo. Nada.
So intensity is an effective way to increase prey detection distance in open environments. This finding was reflected in our results that showed that differences in intensity between Arcapensis in the feignbos and desert biome accounted for greater differences in prey detection than differences in frequency. Also, by emitting very high intense calls, Damarensis was able to achieve similar detection distances to those of archipensis in the desert, despite their use of higher frequency calls. However, intensity is also energetically costly. So bats decrease the intensity of their calls during emergence, which chances of prey capture were slim compared to a foraging area. In addition, high source levels in Damarensis was the exception and not the norm. In terms of frequency, the use of lower frequency echolocation calls in compensis did allow them to achieve greater detection distances, as proposed by the foraging habitat hypothesis, without having to emit those energetically costly echolocation pulses above 130 decibels. However, these greater detection distances could be achieved by emitting higher intensity calls as well, suggesting that lower frequency calls are not a prerequisite for desert survival. Therefore, the lower frequency calls observed in archipensis in the desert could have evolved for another purpose. So, according to the acoustic communication hypothesis, frequency is important for species discrimination and therefore potentially communication. So even though low frequency echolocation pulses in the desert did increase prey detection distances, they could have also evolved for unambiguous intraspecific communication. With those conclusions, I come to the second part of my presentation, where I explore the stipulation made by the foraging habitat hypothesis that proposes that the variation we observed in the acoustic parameters of both frequency and intensity in archipensis in the desert and fambos biomes is actually the result of ecological selection. So ecological selection simply means that variation arises as an adaptation to local environmental conditions, including things such as weather, soil conditions, and in the case of the foraging habitat hypothesis, level of environmental clutter. However, as biologists, we know that variation can also be a product of behavioural flexibility. So behavioural flexibility is defined as the ability of an animal to change its phenotype without selection or genetic variation. An example of behavioural flexibility in bats in echolocation is the changes that they make to their acoustic parameters when they approach a target. Another prime example is the lombard effect. So the lombard effect is simply the tendency of an animal to increase the amplitude of their calls when calling in loud noise to enhance their audibility. So this was observed in the pale spear-nosed bat. They raised the amplitude of their calls, increased the duration of their calls when presented with increasing noise levels. So I wanted to know, are the recorded differences in frequency and intensity between desert and fambos archipensis a result of ecological selection, as was proposed by the foraging habitat hypothesis, or are they a result of behavioural flexibility? I therefore conducted a flight room experiment in both the desert and fambos environments. So bats in both biomes were made to fly in a level of clutter representative of their own biome, as well as a level of clutter representing the other biome. If observed differences are due to behavioural flexibility rather than ecological selection, as proposed by the foraging habitat hypothesis, then bats in both biomes should be able to adjust both the frequency and source levels of their calls to achieve similar detection distances when exposed to the same degrees of clutter. I exposed bats in both biomes to a 2-trial flight room experiment. So first they were flown in a clutter flight room that simulated the fambos biome. And then they were flown in an open flight room that simulated the desert environment. Example of a bat flying in the cluttered environment. So in the desert, bats in the cluttered flight room echolocated at 70 kilohertz. But when they were flown in an open flight room, they echolocated at 73 kilohertz. This is opposite of what you would expect. And this is because something very interesting happened here. Bats didn't actually lower their frequencies of their calls in the cluttered flight room. Instead, they shifted where they put most of the energy of their calls from this constant frequency component known to be used for prey detection to this frequency modulated components thought to be important for ranging. In the fambos, bats did not have the same response, and this is probably because they're used to flying in a highly cluttered space. There was no differences in the frequencies between the open and cluttered flight room. So the large frequency differences between Archipensis in the desert and fambos are likely the result of ecological selection rather than behavioral flexibility. And this makes sense when you consider the echolocation system of high-duty cycle bats. So during flight, high-duty cycle bats emit lower frequency pulses, compensating for Doppler shift effects. As a result, the frequency of the returning echoes fall within a frequency range known as the acoustic fovea. And this is a specific region of the auditory system that is sensitive to a unique frequency range. This region is associated with adaptations ranging from features in the cochlea through to the auditory cortex and the inferior cochlus in the brain. Frequency in rhinolophus is therefore highly specialized, may have a large genetic component, and may have a potential fine-tuning component via mother-to-offspring transmission. So small variations in frequency could be explained by behavioral flexibility or even phenotypic plasticity, but it would not explain the larger 10 kilohertz differences in frequencies we observed in Archipensis between the two biomes. And this is because larger shifts in frequency, such as those required to make an ecologically significant difference in detection distance, might render the bat insensitive to its own echoes, because these echoes no longer fall in the frequency band of the acoustic fovea. Frequency, intensity does not have the same biological constraints. In both biomes, when bats were flown in a cluttered flight room, they lowered the intensity of their calls, giving them smaller detection distances than when flying in an open flight room. And this shows that intensity has flexibility. And these lower call intensities in high clutter could be a ways to reduce energy expenditure. But the fact that desert bats still use higher intensity calls with greater detection distances than fambos bats, when exposed to the same level of clutter, suggests that even though call intensity is flexible, it could also display some level of selection. I did want to mention that we did look at other factors that might affect our acoustic parameters, such as gender, morphology, and atmospheric conditions, for which I can say we found no effect. So my findings have provided some important insight into how bats use sonar to interface with their environment, and how the environment in turn shapes these sonar systems. I would like to conclude this presentation with a few words on Capensis and Amarensis and the amazing ability of these clutter forager specialists to inhabit an open desert environment. As we have seen, the harsh open desert environments has shaped the echolocation pulses of both Capensis and Amarensis. And the ability of these clutter forager specialists to inhabit an open environment is a testament to both the acoustic flexibility and constraint of their echolocation system. This echolocation system is marked by an intricate echolocation pulse design, where even small changes to the acoustic structures of their pulses has had significant ecological and functional impacts. So to sum up this presentation, I would like to say that ultimately, our results tell a story of fortitude that speaks to the remarkability of even the most specialized systems to change and adapt. I'd like to thank every one of you for coming and listening to my talk today, and to the presenters and organizers of Bats Without Borders for this wonderful opportunity. Thank you, Rachel, and thank you, Orly. And to my wonderful supervisors who were with me every step of my PhD journey, David Jacobs and Marc Holderied, and to all the support of the members of the Animal Evolution and Systematic Lab. And finally, to everybody else who just helped me in my PhD journey, including my wonderful husband, Jake Sales, and the amazing interns who helped me in the field, Megan and Aurora. Once again, thank you, everybody.
Thank you very much, Nikita, for the great presentation. Really, really interesting. In particular, I like the interaction that you see there between behavioral flexibility and ecological adaptations. Now, I'm sure people are going to have a lot of questions to ask, so please put your question in the chat. In the meanwhile, I'm just going to ask you one question that came into my mind. When you talked about all these shifts in the echological, the differences in there, especially in the frequency of maximum energy, it made me think about what I noticed a lot of the time when recording bats of different species foraging in the same place, tending to slightly either increase or decrease the frequency of the calls to reduce overlap. And I wonder if that played any factor here, that in one habitat, perhaps there's another species with very similar echolocation calls, requiring the bats to change the frequency to not overlap with them.
Definitely, and that's where I brought in the acoustic communication hypothesis, because Ardamarensis and Archipensis have overlapping frequency ranges. So in the desert, they echolocate at 75, whereas in the feignbush, they echolocate at 84. So one of the theories is that they're lowering those echolocation frequencies so there's no overlap. In addition, in the feignbush, there's also Rhinoplophus clavosus, and that echolocates at 93 kilohertz. So there's also a theory that they could have shifted their frequency up a bit as well. So there was an interesting study done by Bastian et al. That looked at this interaction. So I definitely think that that might be one of the reasons for this shift in frequency. So even though that there is a advantage, so it does provide these greater detection distances without having to expend so much energy, it could have evolved for that acoustic communication part of it.
Yeah, really interesting. And it's interesting how so many factors are playing together to really affect the echolocation call structure of bats. I've got here a question from Rich Flight asking, first of all, saying thanks for a great talk, and a few other people said thanks for a very nice talk and fantastic talk. So Rich is asking, do you think your findings would be the same in bats that use FM calls, not just CF calls?
Definitely not. I think that the echolocations are so different. So there was one study that comes to mind that showed that bats actually shifted their frequency under different atmospheric conditions. So I think that FM bats, they don't have this restraint of the acoustic fovea. So I think that in that sense, they're not as specialized in terms of their frequency ranges. So I think that in that way, they're a little bit more flexible than the high duty cycle bats. So I think it's really like studying two different systems almost.
And actually, if we think about it, a lot of them are phylogenetically as well quite distinct, aren't they?
Exactly. So it's completely different ball range.
Yeah, very interesting. I'm just waiting to see if we got any more questions for people. Don't be shy. Please ask any questions you may have. Let me ask you then something else while we're waiting. You mentioned that these are not the only habitats where these bats are found. And I know you mainly focused on their activity in these habitat types. But do you know what is the mechanism behind differences in other parts, or whether it's similar type of mechanisms, or whether they either will be responsible for that?
So Odendell did a study where she did look at it in all the different variant biomes, and she did, this is what spurred the study actually, she did find a correlation between level of clutter in these biomes and the frequency. But in terms of intensity, these are actually the first recorded intensities of high duty cycle bats in the field. So this research hasn't been done before. So I think it would be interesting to look at the reported intensities and detection distances of those other biomes, and then we'd get a bigger picture.
Yeah, definitely. It'll be really interesting to hear that. Yeah, I'm still waiting for people to ask more questions, but I'm happy to continue asking.
The study, the recording the intensities was very tricky, and it was very new. So we had a lot of tinkering and figuring out with the microphone arrays and everything, because like I said, it's only been done in laboratory environments or controlled flight room environments. So that was very interesting, and there was a lot of trial and error in that. But finally we got it right. And I think one of the main things was localizing those bats, because part of localizing calls involves bandwidth. So the higher the bandwidth, the better localization we get. So luckily, both Capensis and Damarensis used those long FM components. So we were able to use those FM components. So hopefully now that we have this technology, more people can jump on board and start to use it to measure the intensities and detection distances of bats, high duty cycle bats in the field.
Yeah, and I really hope people will start doing this so we gain a bit more information beyond just what we usually use, which is just the frequency of maximum energy.
Exactly.
So we got a question here from Alon Velasquez asking, how do you think would this kind of study be translated to the conservation of bats in your study site? And there's another question, but I'll let you answer this.
Well, I think it's good to, I don't know if it really has a conservation impact, except for the fact that we're learning more about these bats. I guess these days a lot can be used for conservation. I think this was more of a behavioral study, but I guess that's an interesting question. I'd have to really think about that. I don't delve much into conservation issues, but I'm open to hear his thoughts about it.
Okay, so I'll continue with the second question that he asked, and then Alon, I'll give you the opportunity if you want to say anything about this. Please raise your hand, or you can say it in the chat. So the second question was, do you know of any articles or study that you can share that are same as yours, but based in different countries or environments?
So there are other ones that look at low duty cycle bats. So Calco and Serlich and Calco, like 2008, they also found that in low duty cycle bats, they had a similar thing where they compensated for frequencies with intensities. There was also one that my colleague did, and he showed that bats in different foraging niches also use different detection distances by adjusting their intensities of their calls. Honestly, there haven't been that many studies done in the field of bats. There have been a lot in the laboratory environment, but yeah, I would mainly look into Serlich and Calco's. That was a big, and my supervisor also did a couple, Holderied and Halverson. That's also a good paper.
Okay, excellent, very helpful. We had another question from Kate saying, could the flight cage itself have acted as clutter?
Yes, definitely. So it definitely acted as clutter. And the way that we did this was we actually did a clutter index where we measured the level of clutter by using a grid. So for every X meters on this grid, we measured a point, and the same as in the field. So we could actually quantify the level of clutter in each one, and we created this clutter index. So they definitely acted as clutter, but the bats in the open environment emitted the same source levels in the field as they did in the desert environment, that is in the flight room. So they didn't really see it as much more cluttered, but what was interesting is the thambus actually saw it as more cluttered. So they definitely had a little bit of a difference in the reaction. But yeah, that was definitely part of the thing, and we try to control for that as best we could.
Yeah, I suppose that's always the problem with any experimental manipulation.
Yeah, that's why I think it's important to do both the field and the flight room experiment, so you can look at both.
I agree. We have another question from Rich saying, naturally, the prey detection range is different in the two environments, but how do the prey species vary? Are the species that they prey on very different?
That's a very good question, and we actually did control for this. So I did light traps in the field, and they had very similar, well, the sizes. So it was not so much as the species that was important, it was the target strengths. So the size in which the sound is insonified for most targets. And that would tell how much they could tell the difference between small, medium, and large prey. So we ensured that these target strengths were the same for small, medium, and large prey. So the range of prey sizes were the same in both the feignbush and the desert environment. And that's how we controlled for it.
Thanks. And another question from Rich is with the arrays that you used, is the triangulation automated or did you have to do it manually? It seems like it will be very tricky math.
My supervisor created a MATLAB script, which was very helpful. The detection distances, the calculation of the detection distances were manual, which was very tedious. But once I got it into an Excel formula, I got that going smoothly. But no, the MATLAB did automate the arrays, thank God.
Sounds very helpful. And we have a question from Jack Hooker asking, do you think that different populations of the same species could have evolved different levels of behavioural plasticity in their calls across their range in Southern Africa?
Oh, definitely. I definitely think that they could. Behaviour, as we've seen, varies from population to population. So yeah, I think that's a definite possibility.
Okay, excellent. And we have Neil Aldridge asking, slightly related to Richard's question regarding prey. Do you know if prey population density is different between the habitat? Could it be a factor?
We don't know that. We only know the density of feignbush from Jacobs et al., but that population in the desert is very understudied. I had to spend like a month out there just to find the foraging area where they foraged because there's so little vegetation. So I had to set up a bunch of different recording sites all over the desert and I got very lost. But yeah, that's definitely something that I think should be investigated, as well as like the previous question, which species exactly are they catching?
Yeah, definitely to link it to functionality. Well.
Exactly. But I think that's a whole nother like master study on its own.
There's always more research to be done.
Yeah.
We have a question from Javier Juste asking, do you think that individual inherited genetic variation or, do you think that individuals inherited genetic variation or on the other side, learning processes could play any role in bat responses?
I think that it can be a little bit of both. So like I was saying about the frequency, there has, in terms of small changes in frequency, there is both a learned component and an inherited component from mother to offspring transmission. In terms of intensity, I also think that there could definitely be a learned component. And I think that's why bats reacted differently in the two different flight room situations, in the feignbush and the desert. So it's kind of like culture, you know, like you used to one thing and that's why you react a certain way. So in the desert, one of the cool examples of this is that they weren't used to that clutter. So they actually shifted where they put most of the energy into that frequency modulated component to give them that increase in ranging. Well, potentially.
Yeah, really interesting questions here from everyone, and yeah, interesting different perspectives on how things are happening and how things come into play. I was wondering if anyone has any few last questions that they wanted to ask Nikita. Okay, so if we don't have any questions to ask, what I would say is I'll ask everyone if we can thank Nikita for the excellent talk. And
Thank you.
Name of everyone who's got the microphones muted. And just to mention, if people are interested, Bats Without Borders is hosting webinars, these kind of Winged Wednesday webinars on quite a regular basis. So please check social media sites to find out a bit more. And if you want to help Bats Without Borders and maintaining these kinds of webinars, you can donate, and I shared the link in the chat. So thank you very much, everyone. And especially thank you, Nikita, for a great talk.
Thank you for organizing it.